Hello, we're back to the practical class part 2 on karyotype analysis and, uh, and we will be discussing in this session the description of the structural chromosomal anomalies through the karyotype analysis and the second method for chromosome study using molecular cytogenetics techniques. Just to refresh your memory with the structure of the chromosomes, there are three types of chromosomes, the metacentric, submetacentric, and the acrocentric. And these are, التقسيمة دي is based on البوزيشن of the centromere. لو السنترومير كانت central بيبقى metacentric والsize of the P arm is almost or slightly shorter than the Q arm or the long arm. في حالة السبميتاسنتريك كروموسوم ال P arm بيبقى much shorter than the Q arm فالسنترومير بتبقى shifted more towards the P arm. The acrocentric chromosome, the centromere is peripheral. There is no P arm, only a Q arm is present. And instead of the P arm, there are satellites attached to the centromere by a stalk. And it's important to mention here that the satellites do not have important single copy genes, but all satellites of all acrocentric chromosomes carry genes that code for synthesis of ribosomal RNA. So there are a large number of genes that code for ribosomal RNA. And so this means that the satellites, even if they are lost, they will not affect the genome balance, and it will not cause any phenotype abnormality. How can we describe a chromosomal locus? Using a banded karyotype, the G-banded karyotype, which is the standard preparation of the karyotype, as we described in part 1 of this practical class, each arm of the chromosome is divided into regions, which may be 2 or more, and the regions are divided into bands. The numbering of the regions starts from the centromere and goes towards the peripheral end of the P and the Q arm, that is the telomere. And the same thing, the numbering of the bands starts from the centromere towards the telomere as well. And if the karyotype is formed of long chromosomes, they will be subdivided further. The bands will be subdivided further into sub-bands. So this diagram here can illustrate this X chromosome diagrammatic representation, and you can see the Q arm is subdivided into 2 regions. Region 1 is the closer to the centromere, and then the region 2 is subdivided into 2 bands, 1 and 2, and you can see here that the region band 2 is subdivided into subbands 1 and 2. So if we want to determine the locus of XQ22.1, this means that this locus is present on the long arm of chromosome 2 in region 2, so it is in this region here, in band 2, so it is somewhere here, and it is in subband 1, so this locus is present here. The locus is used for mapping of the gene loci. So if you want to describe where the gene lies on a chromosome, you describe its locus, and through the G-banded karyotype, you determine the locus position according to the number of the chromosome, the region of the chromosome, the band, and sometimes you put a dot after the band, and then you mention the subband. So you never pronounce it as XQ22.1. This is wrong. You pronounce it as XQ22.1, which means region 2, band 2, subband 1. This is the master flow chart of all the chromosomal abnormalities. Always go back to this classification so that you can remember how different chromosomal abnormalities are classified. Today we are concerned with the structural chromosomal abnormalities, and we will go through the different types of these abnormalities, and we will have some karyotype examples on them. I will just mention to you here that the marker chromosome is a chromosome which is structurally abnormal, but you cannot identify with the G-banded karyotype its exact nature. It results from translocations between different chromosomes, and so the exact nature of which is not known, and therefore it is called as a marker chromosome. This table here shows the symbols that are described according to the International System of Cytogenetic Nomenclature, and how you describe a karyotype with a structural chromosomal abnormality. So you can go through these before describing the different karyotypes. I would just want to give an example. Again, you mentioned first the number of the chromosome, comma, the sex chromosome complement, comma, and then the structural abnormality type. If there is loss of genetic material, this is a deletion, so you put the symbol del. And then you put where the deletion is present. So here it is present on chromosome 3, in the long arm, in region 1, in band 3. So this means it's a terminal type. If the break is single, one break only, كسر واحد في الكروموسوم، والجزء اللي بعد البريك ده، الجزء اللي بعد الكسر ده is lost. So where this break lies, it lies in region 1, band 3. So this means in distal اللي بعد الجزء ده, locus ده, will be absent, will be lost due to the deletion. Okay? Ring chromosome, which is another type of deletion, is given the symbol R, and if one of the two X chromosome is a ring chromosome, so this is written as 46, X ring X, or sometimes it's written as XX and then comma RX. The inversion is two breaks, and the part in between the two breaks rotates so that it becomes upside down. تتقلب وتبقى في الاتجاه المعاكس. طيب ازاي هنوصفها؟ السيمبل is inversion, three first symbols of the word inversion, inv. So it will be a 46, the number, total number of chromosomes is not changed with inversion. XY, this is the sex chromosome complement, and then the symbol of the structural abnormality, open brackets, where the inversion is present, it's here in chromosome 5, long arm, region 1, band 1, and this is the first site of the break, ده أول كسر موجود. 2, البارت اللي حصل لها inversion lies between the two breaks. This is the first site of the break, ده أول مكان حصل فيه كسر، ده تاني مكان اللي هو region 3 band 1. اوكي؟ لو نلاحظ هنا الانفيرشن موجودة في one arm، يبقى معنى كده ان هي it's on one side of the centromere، فعشان كده اسمها paracentric inversion. لو أخدنا دي الاكزامبل التاني بتاع الانفيرشن، برضو 46XY inversion موجودة في كروموسوم 7 short arm، first البريك سايت في الشورت arm region 1 band 2. السكند بريك في اللونج arm of the same chromosome region 2 band 2. بما ان التو بريكس حصلوا on one in the P arm and one in the Q arm، يبقى الجزء اللي هيحصل له reversal of direction هيلف ويرجع يلزق تاني includes السنترومير. يبقى في الحالة دي ال type of inversion is pericentric. Peri means around، يعني around the centromere، يبقى السنترومير موجود في الجزء اللي حصل فيه الانفرشن. تمام؟ طبعا من هنا كده لو انا طلبت منكم انكم تقولوا لي انهي من الاربع انواع دول اللي بيبقوا balanced، يبقى انتوا عارفين طبعا ان الانفيرشن ما فيهاش loss or gain of genetic material، فبتبقى balanced karyotype، ولكن ممكن تأثر على الجين expression وممكن تأثر على الفيوتشر generation لواحد او واحدة carrier for inversion. بالنسبة للترانسلوكيشن احنا عارفين احنا عندنا two types of translocation. روبرتسونيان translocation بتحصل فقط في الاكروسنتريك كروموسومز اللي هما كروموسومز 13 14 15 21 and 22. في الحالة دي البريك اللي بيحصل في التو كروموسومز بيحصل at the centromere، فكل الكيو ارم بيلزق في الكيو ارم بتاع الكروموسوم التاني، يبقى عندي one single كروموسوم كبير لكنه بيحمل carries all genetic material of both chromosomes. وعشان كده احنا بنقول هنا ان هي ادي كيو ارم المكان الترانسلوكيشن between 13 and 14 والسايت اوف ذا بريك كيو ارم region 1 band 0 لانها عند السنترومير نفسها. It didn't go past the centromere. نفس الكلام بالنسبة للبريك where in chromosome 14. النوع التاني من الترانسلوكيشن اللي هو الريسيبريكال ترانسلوكيشن، وده اللي هو بيحصل في الميتاسنتريك والسب ميتاسنتريك كروموسومز. السيمبل بتاعه بيبقى تي، ونفس الكلام هنا بنحط اللي هو ال exchange of segments of chromosomal segments هنا حاصلة ما بين كروموسوم 6 and كروموسوم 9. جزء من البي ارم او البريك اللي حصلت في البي ارم اوف كروموسوم 6 again region 1 band 1، والجزء بتاع البريك اللي حصل في كروموسوم 9 موجود في اللونج ارم region 2 band 1. اوكي، يبقى كده الاكسشينج between C 6 كروموسوم 6P and 9Q. الايسوكروموسوم السيمبل بتاعه I، واحنا عارفين ان الايسوكروموسوم ده يعني ان الكروموسوم بيبقى formed of two similar arms, either the two P arms or two Q arms, because centromere during the anaphase the centromere was split horizontally instead of vertically, فال two arms of the chromosome is the same. فهنا هيبقى عندي 46, X, one X normal and the other is an isochromosome X involving the QR. The duplication is the gain of a chromosomal part due to because of unequal recombination during crossing over. فبيبقى في one segment of the chromosome is duplicated. It means instead of having one copy of it we have two copies. فعشان كده الاكسشينج هنا مش زي في الترانسلوكيشن is between non-homologous chromosomes. دول non-homologous دول non-homologous، لكن هنا بيبقى the unequal recombination في crossing over between homologous chromosomes، وعشان كده بيبقى السيجمنت is duplicated. اوكي، طب ليه برضو بنوصفها هنا بالطريقة دي؟ ال duplicated segment is between the long arm region 1 band 2 and the second break is at region 2 band 2. Okay, and it is in the long arm of chromosome 7. The most common type of structural chromosomal abnormalities بنشوفها بتبقى الترانسلوكيشن. الترانسلوكيشن دي بتعتبر the commonest type، وخصوصا في الكانسر، في الهيماتولوجيكال malignancies البلاد كانسر زي leukemias and the lymphomas. Okay, this is an example of what? It's a reciprocal translocation. As you see here, break there, and exchange of segments. So you will have balanced translocation between these two chromosomes. No gain, no gain and no loss of genetic material. Now, the person with a balanced translocation will have a normal phenotype, but they will have problems when they form gametes. Some of their gametes will be unbalanced. They can take one normal chromosome and a translocated chromosome. So there will be an extra segment of this chromosome and a missing segment from that chromosome. And so the problem will be in the product of conception. يحصل بقى abortion, يحصل miscarriage, أو يحصل abnormal children with congenital abnormalities. Okay, let's try now to describe this karyotype. The arrows here show that there are differences between the two homologous chromosomes, right? So we will follow the same scheme. Number, so count the number of the chromosomes. You have a pair of each chromosome, so the number is 46, comma, what is the sex chromosome? It is XY, so this is a male. Now we look for the abnormalities that are present. In order to look if there is no numerical abnormality, then look for each pair of chromosome. هنبص على كل اتنين ونشوفهم شبه بعض ولا لا. طب لما نبص على chromosome 4 هنلاقي there is part of the chromosome, the terminal part of chromosome 4 is absent, right? طب نبص راحت في حتة تانية، هنكمل برضو نبص على each pair of chromosomes زي بعض ولا لأ. هنلاقي ان الجزء ده اللي هو اهو ادي الدارك باند وادي دارك باند كمان موجود، وهنا انتهى الكروموسوم ده هنا، وادي التو دارك باندز بتوع كروموسوم 4 كيو، يبقى اتنقل وراح عند كروموسوم 11. اوكي، وغالبا التيرمينال بارت بتاعت الشورت ارم اوف كروموسوم 11 اللي هنا اتنقلت وراحت عند كروموسوم 4. يبقى this reciprocal translocation between 4 and 11. What about chromosome 9؟ المفروض ان في جزء في ال P arm في dark band. It's absent from here. يبقى في deletion في ال P arm of chromosome 4. So now let's describe this karyotype. Number is 46. This is a male. عنده reciprocal translocation between 4 and 11. وعنده deletion في short arm of chromosome 9. Deletion in 9p. Okay? طيب, mention if this is a balanced or an unbalanced person. هو لو كان بترانسلوكيشن بس كنا هنقول إن هو balanced لأن الكروموسوم 4 and 11 just exchanged segments. But because this person has deletion in the short arm of chromosome P, which was not transferred, exchanged with any of the other chromosomes, يبقى ده unbalanced karyotype. يبقى غالبا هنا هيكون في some abnormalities in this person. I mean phenotype abnormality because there is loss of genetic material from the short arm of chromosome 9. This is another karyotype that we need to describe. Okay, we first look at the number of the chromosomes. Is there a numerical abnormality here? زي ما احنا شايفين there is one chromosome 14. Before I rush and say that this is a monosomy, I have to look if the other chromosome is attached to any of the other acrocentric chromosomes. So you can see here that one of the chromosome 13 is very long. And normally it's an acrocentric chromosome that should not have a P arm. So this means that this is another chromosome and it's actually chromosome 14 being attached to chromosome 13. So it is a Robertsonian translocation which is joining of the Q arm of 13 and 14. Okay? So the sex chromosome here is XX. So the total number of chromosomes will be, you count the bodies. You don't count this as two chromosomes, even though it carries the genetic material of two chromosomes, but because it's a single body, so it is counted as one. So the total number of chromosomes is 45, XX, and this is the structurally abnormal chromosome, which is called Robertsonian translocation between 13 and 14. So this is a balanced karyotype and the person is called a carrier with a Robertsonian translocation 1314. The person will have a normal phenotype, but again may have problems with the gamete formation. Let's describe this karyotype. Again, we look at each pair of chromosomes to see if there is a change in the number of the chromosomes. So we have a single chromosome 21. Before we decide that this is a case of monosomy, يعني a single copy of chromosome 21, we have to check if there is a Robertsonian translocation that involves chromosome 21. So we look at the acrocentric chromosomes اللي هما الجروب ده والجروب ده. ادي chromosome 13، ادي السنترومير هنا والسنترومير هنا، يبقى this is okay acrocentric chromosome. نيجي هنا على chromosome 14، ادي السنترومير هنا، there is nothing above the centromere or the satellite، فبالتالي this chromosome is okay. Let's look at this chromosome. Well, it should end here. هنا السنترومير يبقى في جزء زيادة، الجزء ده بيشبه جدا chromosome 21، بيبقى عنده dark band just next to the centromere وبعدين a light band. يبقى ده is the second copy of chromosome 21 in the form of Robertsonian translocation 1421. Okay? And the rest of the chromosomes are okay. كل الهomologous chromosomes شبه بعضها and the sex chromosome is XX. So describe this karyotype. Try to pause. نعمل pause ونحاول احنا ندescribe الكاريوتايب and then we check. تمام؟ Okay. So it is described as 45. اتفقنا ده هيتعد as one chromosome, counted as one chromosome. So it's 45 XX, and there is a translocation, the Robertsonian translocation between 14 and 21. Now, let's answer this question. Is this female phenotypically normal? What do you think? She has a balanced karyotype because the missing chromosome 21 here is attached to chromosome 14. Is there any genetic material lost or gained? The answer is no. So she is a carrier, just a carrier of an abnormality in her chromosomes. So she is having a normal phenotype because of a balanced karyotype. And the second question, what might be the reason for asking her to do a karyotype? نتوقع ليه طلبنا إن احنا نعمل karyotype for this female. هناخد السلايد التانية وهي دي اللي will answer. We will go to the other slide which will answer this question. Now this female has got, instead of 2 copies of 14, 2 copies of 21, which is the normal chromosomes, she has 114, 121, and 1421 attached together, right? As a Robertsonian translocation. So what will her gametes carry? Now, she is supposed to have these 3 chromosomes divided into 4 gametes, right? And so the probability of the segregation, نقل الكروموسومز دي إلى الفور gametes, الفور ova that she will produce, the probability will be as follows. يا إما the gamete will have a normal 14, a normal 21. This will be a completely normal gamete and will have, if the ovum is fertilized with a normal sperm, هيبقى عندي completely normal baby. أو إن الأوفم تاخد ال 14 21 attached together وتبقى برضو balanced بس هتبقى carrier like the mother ويبقى برضو genetically هتبقى normal وهتدي لي normal baby carrier of a Robertsonian translocation. أو إن الجميت ياخد ال Robertsonian translocation ده وياخد معاه الكروموسوم 21. Okay? فهنا الجميت ده هيبقى عنده كروموسوم 14 1 copy، كروموسوم 21 2 copies. فلو حصل له fertilization by sperm ساعتها هيبقى فيه how many copies for chromosome 14؟ 2 copies of chromosome 14. The zygote will have 2 copies of chromosome 14. And how many copies of chromosome 21? 3 copies of chromosome 21. 2 from the ovum of this carrier female and 1 from the sperm. مادام في 3 copies of 21 يبقى the phenotype of the baby will be Down syndrome. Okay? أو إن الجميت يبقى عنده just 14 and missing 21 يبقى monosomy and therefore it will not survive. أو يبقى عنده only 121 and monosomy 14 فيبقى بالتالي برضو will not survive. أو يبقى عنده extra copy of chromosome 14 لأنه أخذ ال 14 وأخذ ال Robertsonian translocation which contains 14. فلو ده حصل له fertilization by a normal sperm ساعتها هيبقى trisomy 14. واحنا اتفقنا ان الترايسومي اي حاجة of the autosomes except 21 و 13 و 18 because these are the chromosomes which carry the smallest number of genes. Other than this هيبقى lethal وهيحصل abortion أو أصلا الجميت cannot be fertilized. إذا نتوقع إن this female did a karyotype analysis because she may have had a Down syndrome and the Down syndrome was of translocation type. فبالتالي عايزين نعرف if she is a carrier of translocation type then she may get another child which will be also Down. Okay? Or she may have repeated abortions because of this abnormal gametes, or she may be suffering of miscarriage with abnormal fetus. تمام؟ يبقى ده هو هيكون السبب ده اللي هيجاوب لنا السؤال why did this female do a karyotype even though she is looking completely normal. طيب عايزين نعرف هي هتسألنا لو هي كانت عاملة الكاريوتايب because she had a down baby of translocation type فإحنا عملنا الكاريوتايب بتاعها ولقيناه that actually she is a carrier for chromosome 1421. فهتسألنا إيه قد إيه فرصتي إن أنا أجيب another down baby. Let's calculate her risk to get another down baby. دلوقتي أنا دول will be lethal يبقى مش هيدوني living baby. خلينا concentrating على اللي هيديني a normal baby. الchances إن هي تبقى 2/3 يديني normal baby بكاريوتايب يا إما completely normal يا إما كاريوتايب carrier of translocation. تمام؟ يبقى 2/3 will be completely normal and 1/3 will be a down baby. 1/3 يعني 30% إنها تجيب إن الكونسبشن بتاعها إن الفيرتيلايزد أوفا بتاعتها في 30% of the cases إنها تبقى معاها extra copy of chromosome 21. ولكن بسبب إن حتى في الداون pregnancy بيحصل very high بتبقى at a very high risk إن يحصل abortion ب 50 to 60% of the cases. تمام؟ يبقى في الحالة دي recurrence risk بتاعها إنها to get another down baby is not 30% but less because she can have losses of the down pregnancy. فبتبقى المعروفة النسبة المعروفة of recurrence لو هي carrier إنها تبقى from 10 to 15%. But she has a risk of 10 to 15% to get another baby with Down syndrome. طب لو كانش هي لو أنا عملت الكاريوتايب وهي كانت completely normal and she doesn't have a translocation الريسك بتاعتها بتقل كتير جدا عن 10 to 15% وتبقى زيها زي الريسك of any female who got a down baby وبتنزل ل 1% لو ما عندهاش if she's not a carrier of the translocation بتبقى الريسك only 1% like any female who has got first child as down يبقى to get another down baby is only 1%. This is the second type of chromosomal abnormality which is called the deletion. الديليشن معناها أني بفقد جزء من الكروموسوم loss of part of chromosome نتيجة لبريك حصلت في الكروموسوم كسر في الكروموسوم وجزء من الكروموسوم اتفصل وما لزقش did not join back and was lost. فلو البريك is at one end of the chromosome سواء في البي أو الكيو والdistal part is lost بسميها النوع ده بسميه terminal deletion. If you have two breaks in the chromosome and the middle part between the part between the two breaks gets lost, did not join back والكروموسوم بيبقى أقصر مما كان وال two breaks join together and the middle part is lost دي بسميها interstitial deletion. And if there is a terminal break at the P arm and another at the Q arm and the ends stick together الكروموسوم يتحول شكله بدل ما كان straight linear يبقى شكله like a ring, and this is the ring type of deletion. Describe this karyotype as normal. Number, we can see here that there are two pairs of each chromosome and XX for the sex chromosome. So the number is 46, the sex chromosome is comma XX, and now we compare each pair. كده هنبتدي نقارن كل اتنين شبه بعض ولا لا. لو بصينا هنا على الكروموسوم 5 هنلاقي ان في حتة منه طايرة lost اللي فيها الدارك باند اهي terminal part of the P arm of chromosome 5 is not present, absent, is broken, is lost. يبقى there is a deletion in the P arm of chromosome 5. Okay, let's continue and screen the rest of the chromosomes. Maybe we find another abnormality. Well, I cannot see any other abnormality, so description of this karyotype will be here. The only abnormality is the broken terminal part of chromosome 5, deletion 5p, and so the description of the chromosome هو represented هنا بالايديوجرام. يبقى شفنا هنا ايه فايدة الايديوجرام؟ ان انا بقارن الكاريوتايب بتاعي النتيجة بتاعتي اللي بتطلع من الكروموسوم study. I compare it to the ideogram of the chromosome that has got the structural abnormality to determine where the break happened. يبقى البريك اهو موجود هنا، ادي الدارك باند اللي موجودة بعد السنترومير. اوكي، يبقى this is a 46XX deletion 5p. ده بيؤدي إلى سيندروم اسمه كري دوشا. كري دوشا يعني the cry of the cat, because this baby with deletion of the 5p will have sound of the cry is the same sound like the sound made by a cat. the mother will complain. هتيجي الأم تشتكي إن صوت عياط البيبي غريب وإنه بيبقى صوته زي القطة بالظبط. الطفل ده اللي عنده deletion of the 5p بيبقى راسه صغيرة، يعني البرين بيبقى smaller, the brain is smaller than normal. بيبقى عندهم كمان low set ears، بيبقى عندهم thin upper lip، بيبقى عندهم the anteverted nose، يعني بيبقى فيه some features بيبقوا كلهم يعني فيهم features في الفيس which are similar، لكن أكتر حاجة بيبقوا إن هما طبعا بيبقوا mentally retarded because of the small brain. دي أهم defect بيبقى موجود عندهم. Let's describe this karyotype. The number of chromosomes is two pairs of each chromosome, so it is 46, and comma, we have, this is the sex chromosome is one normal X and the other X chromosome is looking like having the shape of a ring, and this is described as 46XX ring X, and sometimes we just write one X and the second X as an R X. Okay, So what happened here is that was a broken end from the P arm and a break in the Q arm, and the two ends fused together to form a ring. Okay? This case can present, of course, this is an unbalanced karyotype, and it can present as what? Turner syndrome. It is one of the types of Turner syndrome. How about this karyotype? Again, we have pair of each chromosomes. So the number is 46. We have two X chromosome. But if you compare this, this is the normal X chromosome. You can see the ideogram next to it, which shows this is the dark band here, dark band there, light band, a thin dark band at the end of the P arm, and there are several dark bands, mainly looking like they are three dark bands in the long arm. So this is the normal X chromosome. P arm, it's a submetacentric tall chromosome or intermediate in size, because usually the X chromosome is put with this group C. Now, if you look here at this chromosome X, you find in الارم ده is like a mirror image of this arm. It looks exactly the same. والجزء ده هو الكيو arm. فلما يبقى نفس الكوبي، نفس الكيو arm هنا وكيو arm on the other side of the centromere, this is called what? Isochromosome. So the description here is a 46XX, I for isochromosome, open the brackets, which chromosome is affected? It's the X chromosome. Which arm is affected? It's the Q arm هو اللي موجود عندي في الأيزوكروموسوم ده. What is the clinical diagnosis of this case? Is this balanced or unbalanced? Of course, it is unbalanced here, right? And what is the clinical diagnosis of this case? This will present as a case of Turner syndrome. Okay? Now, mention the type of this structural abnormality and its effect. So what can we see here? Again, we will compare the each of the pairs of the chromosomes. So far, each two chromosomes are looking okay, similar. We come here, there is a problem with this chromosome. You can see here this short arm of the chromosome 16. This is the long arm of chromosome 16. If you look at this, you will find the dark band here has moved up, right? And this is pretty similar to here. So what has happened in chromosome 6, you think? The rest of the chromosomes are fine, and it's a male XY. So what happened here is just reversal of the direction of the chromosome segment. So the Q arm moved up in the P arm, and the P arm went down in the Q arm. So this is a case of inversion that is affecting the chromosome 16, and it involves the P arm and the Q arm. P in region 1, 3, so the break happened here, and the break happened in the Q arm here, region 2, band 2, and then the segment was reversed. And if it is involving which the P arm and the Q arm, so the centromere also moved in the segment that is inverted. So the type of inversion here is pericentric inversion. What are, so we come here to the end of the examples of the karyotype, and I would advise you to go through the presentation and again after you finish, and to test yourself on writing the description of the karyotype through making a pause during the presentation, and then you check on yourself. The second session of this practical class is to know about the other methods that are used for chromosomal analysis. And just remember that the G-banded karyotype is the standard method and the cheapest as well. يعني هي بتعتبر أرخص حاجة وبتعتبر كمان هي الجولد ستاندرد. لكن في طريقة تانية اللي هي المولكيولر cytogenetics. المولكيولر في very nice video on the YouTube على المولكيولر cytogenetics. I advise you to watch it. It's very simple, حاجة سهلة وبتوري different types of molecular cytogenetics, and it also describes the principle, الأساس behind molecular cytogenetics. Here I will briefly explain the principle. إن احنا بيبقى عندنا جزء من ال DNA. This is a segment of the DNA or a segment of the chromosome. طبعا احنا عارفين إن any part of DNA, the DNA is double stranded, and the two strands are held together by the base complementarity, the purine and the purine and pyrimidine bases. Always A is pairing with G, and always T is pairing with T with A, and always C with G. Okay, so if we want to detect the presence of this segment, we will prepare a probe, and this probe is going to be complementary to the to one of the strands of to one of the two strands of the DNA, and and this part of the DNA during the preparation will be denatured, which means that by heating, the two strands will move apart from each other by breaking the hydrogen bonds between the bases of the DNA. And when you break this hydrogen bonds, when you cool down the temperature, the probe can stick again by base complementarity. The probe, because it is smaller than the other strand of the DNA, it will easily bind by base complementarity again to the complementary segment of the DNA. And when it binds to it, if you wash during the preparation, it will remain stuck to this part of the DNA, and the probe is labeled with fluorescence. بيبقى attached لها a fluorescent dye. So when you examine under a microscope, this fluorescence will shine, and then you will know that this part of the DNA is present, and you can even count the number of signals, and it can give you information about what segment of DNA you were looking at. Let's take consider ايه الاستخدامات بتاعة molecular genetics، ايه الapplications of molecular genetics. Molecular genetics, even though is more expensive, لكنها is rapid method of testing, and يعني it can you can just detect a certain abnormality. يعني مثلا if you use a locus specific probe, يبقى you will not be looking at the whole set of chromosomes, but you will be concentrating on a certain abnormality, like for example testing for translocation, and we will see how. Or you can be looking for a certain locus where a gene is present, and you use a probe for this gene to detect the number of copies of this gene, so it can diagnose amplification of the gene, increased copy number, or deletion of the gene, which means loss of this gene. The probes can be directed to the centromere, so and it can therefore be used for counting the number of the chromosomes. Of course, each chromosome has a specific centromere sequence, so you can use a chromosome centromere specific to detect aneuploidies, and this is a very quick method for diagnosis of aneuploidies like Down syndrome or trisomy 13 or 18 or sex chromosome abnormalities, especially in prenatal diagnosis, because it can give a very quick analysis. You can have the probe directed to a locus like here or centromere, or you can have the probe which can cover the whole chromosome, in which case the whole chromosome will light up with fluorescence, and so it is called chromosome painting, and it is very helpful to detect structural chromosomal abnormalities. If you use probes that can cover the whole set of chromosomes, and they all can be seen as the karyotype, this is called a spectral karyotype, and it's usually used to detect complex abnormalities when there are several abnormalities present and it is difficult to detect it with a normal standard karyotype. Like in cancer, spectral karyotype here will be very useful to determine these complex karyotypes. The final type of the molecular cytogenetics is called comparative genomic hybridization, and from the name comparative means you are comparing two things. You are comparing two genomes, right? And it is based again on hybridization, the same principle, نفس الطريقة اللي احنا اتكلمنا عليها في السلايد اللي فاتت، ان احنا we will be using this principle of base complementarity, and this comparative genomic hybridization بتبقى بالظبط زي الوزنة، كأنك بتوزن اتنين DNA، اتنين different genomes، وبتشوف أنهي فيها زيادة أو نقص. يبقى بتشوف الأجزاء اللي فيها deletions والأجزاء اللي فيها amplification all over the genome. تمام؟ هنشوف صور لكل من الإكزامبلز دي توضح لنا الطرق المختلفة للmolecular cytogenetics in future slides. شايفين هنا أنا بستخدم probe علشان for the locus on long arm of chromosome 9, and the locus is called ABL, and another locus on long arm chromosome 22 اللي هي the BCR locus. Okay. لما هستخدم أنا two probes, one labeled red, one labeled green. يبقى لو هي normal cell المفروض إن أنا ألاقي two spots green و two spots red because I have two chromosomes 9 and two chromosome 22. This is in the normal case. لما أبص this cell هلاقي عندي one spot red، يبقى one normal locus for 9 chromosome 9, one locus green, this is a normal chromosome 22, and then I have merging or mixing of the two fluorescent spots together. يبقى معنى كده ان هما لزقوا في بعض. This means that these two spots are overlapping. يعني معناها ان في translocation. يبقى this can diagnose if I find this signal like here and here and here and here and here, this means that there is translocation between 9q and 22q، وده اللي هو معناها ان two loci have come close together، فبسميها BCR-able coming together. That is a very famous translocation happening في leukemia، واسم الكروموسوم لما بيحصل translocation between 9 and 22 بيتسمى Philadelphia chromosome. It's very quick, it's very easy, and you can diagnose straight away that this patient is positive for Philadelphia chromosome, and this can diagnose in الحالة دي عندها myeloid leukemia. Okay? الميزة الثانية the other advantage of this technique, which is called the fish is fluorescent in situ hybridization. هنا I used a probe for a locus, two probes for two different loci, where الميزة هنا إن I don't need to grow the cells in vitro and to divide the cells. I can do it on interphase cells. يبقى هنا ده nuclei of interphase cells, not on metaphase chromosomes. فبتبقى very rapid, very quick. Within 24 hours we can diagnose the case. من الاستخدامات التانية other applications for the fish, again fluorescent in situ hybridization, إن أنا أشوف amplification of a gene. Amplification means increased copy number of the gene. Normally we have 2 copies of the gene, one on each of the 2 homologous chromosomes. This is the normal copy number in a cell. One maternal and one copy paternal. لو حصل amplification of the gene يعني increase copy number of the gene زي ما بيحصل في حالات الكانسر مثلا, يبقى ساعتها I can check this by using a probe against a locus of this gene. Here we have the HER2 gene. It's a gene that has a role in cell division. So if the number of the copy number is increased, يبقى this can be detected by the fish. Again, it can be detected on non-dividing cells in interphase nuclei. And you can see here I'm using one probe for this gene, and you can see that the spots here are more than 2, and big spots means that there is increased copy number of the gene. يبقى this fish for HER2 diagnose that the gene is amplified. Okay? This gene gets amplified in cases of breast cancer. The gene amplification can also be tested in metaphase chromosomes. So this time it's not interphase nucleus but or interphase cells, but first we prepare the metaphase chromosomes like in the karyotype, and then we use the probe for another gene to test for amplification copy number of this gene. If I find just a single copy, now this time I don't see a single copy. You know why? Because in a metaphase chromosome, it's formed of 2 chromatids, right? So this here, this is one chromosome, but has 2 chromatids. So I see 2 tiny spots, which means a single copy on each chromatid. This metaphase chrome 1, it divides in anaphase. Each of the daughter cell will have just 1 copy of these 2, okay? So this is a normal copy number. But here you can see all this fluorescent patch is quite big in comparison to this spot. صح؟ أكبر بكتير جدا, واسع جدا. معناها إن في amplification عالية جدا for this gene. يبقى this also another gene which is has a role in cell division. And when it is amplified, this can happen in some types of cancer, and it will be diagnostic for the genetic change of certain cancer types. The third use for the molecular cytogenetics is to detect aneuploidy, which means change in number of the chromosomes. If this is 2 probes, one red for chromosome 21, centromeric probes for chromosome 21 is red, and the green signal or the green fluorescent dye is used for labeling a probe for the centromere of chromosome 13. And here you can see in this cell you have 3 copies for 3 red spots, which corresponds to 3 copies of chromosome 21 and 2 copies of green. So the normal, so this can be confirmed in this karyotype. You have 2 copies of all the chromosomes except chromosome 21 is 3. So this karyotype confirms this fish analysis where there are 3 signals for chromosome 21 and 2 signals for chromosome 13. اوكي. التست ده الفيش ده بيبقى very rapid وبيبقى سهل جدا انه يتعمل للحالات في ال prenatal diagnosis of cases of aneuploidies. Later on it will be confirmed by the karyotype. Okay, so it's like a preliminary test because it's easy and quick. اللي فات ده احنا اتفقنا كان اسمه الفيش fluorescent in situ hybridization for a spot on or a locus on the chromosome. النوع التاني من المولكيولر سايتوجينetics اني ابقى عندي a whole chromosome painting. يبقى انا كده مستخدمه بروب mixture من different loci on the chromosome بحيث وكلهم labeled with the same color. كله green اهو. يبقى like you have painted the chromosome here, for example chromosome 1, you painted all chromosome 1 with green. فيبقى أنا كده عندي two normal copies of chromosome 1. Right? Now the useful the chromosome painting is very useful to detect translocations. بصوا هنا وكمان it can show you if it's a balanced or an unbalanced translocation. This is a metaphase spread. طبعا chromosome painting هيتعمل على metaphase spread ما ينفعش على الانترفيس. يبقى أنا هنا عندي أدي one chromosome تمام. أدي labeled in yellow and one chromosome painting the other chromosome in red. So you can see here there are two yellow chromosomes, one normal red chromosome and one chromosome which has part from the yellow and part from the red. But there is a translocation here present. There is a translocation in this chromosome, and you can easily say if this is a balanced or unbalanced translocation. So what do you think? Is it balanced or unbalanced? What do you think? Well, there is an extra part of yellow here, and so this means that it is an unbalanced chromosomal abnormality. If in the other chromosome you have, if in this chromosome you had a red part, a red locus, it will be reciprocal balanced translocation. But because you don't have in the two yellow a red part from this chromosome, so it means that it is an unbalanced translocation. This is the spectral karyotype. It's a type of chromosomal painting, but here it's applied to the whole karyotype. It is very useful in showing translocations and structural chromosomal abnormalities, especially if they are multiple. And the karyotype will be difficult to interpret. So you can see here that there is translocation between this, and there is translocation here between the yellow and the white. You have a bit of this chromosome here. So this is a complex karyotype, and so a spectral karyotype is useful in testing this complex abnormalities. Usually this kind of analysis is done in cancer because cancer does have a lot of chromosomal abnormalities. The final type of the molecular cytogenetics is called CGH, and CGH stands for comparative genomic hybridization. Why do we call it a microarray? Because here we have the metaphase chromosomes, or the chromosomes, probes of the chromosomes are present. The different chromosomes, the 23 chromosomes, are present on a glass slide, and they are stuck to a glass slide. And then we have two DNA, two genomes, for example, one from a tumor and one from a normal DNA. Both are labeled with different dyes, one red, one green. And these two genomes will be allowed to hybridize to the probes that are present on the this glass slide, this array. Okay, so if they hybridize and to the probe and there is no and there is a yellow, the signal appears yellow, which is a mixture of the red and green. This means that both of them are balanced. There is no gain or loss on these chromosomes, right? So this is chromosome 3, 4, 5, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and so on. All these here have almost a balanced genetic material. If you come here, you can see that there is excess of the red dye. So this means there is gain of genetic material in the tumor. And when you see only a green dye present in certain part of the chromosome, this would mean that it is deleted in the tumor, and that's why the signal of the normal is only present. طبعا ده بيتعامل بإيمج analysis، يعني you cannot, it's not done manually, you cannot just, unlike the other types which you judge by self-examination. لكن هنا بتبقى الحكاية بتتعمل بإيمج analysis and weighing the two intensity of the intensity of both fluorescent dyes، ويطلع plot بالمنظر ده يقول لنا إذا كان في gain or loss on a certain chromosome. Okay, so here is the DNA different. ناخد بالنا هنا هو يعني دي another picture عشان بس ايه ما نبقاش متلخبطين. احنا فوق قلنا ان red is tumor and the green is normal. هنا متلخبطين بس ده ما يفرقش معانا، احنا بس عايزين نقول ان الكونسبت بس two different DNA two different genomes، واحد normal as reference وواحد بتاع التيومر. بيحصل لهم hybridization على السلايد اللي فيها probes من different chromosomes. تمام، وبعدين أوزن التو dyes. لما الاقي excess of the tumor DNA يبقى عندي amplification، لما الاقي excess من النورمال DNA يبقى عندي deletion في التيومر. يبقى انا كله النورمال ده بستخدمه as الريفرنس بالانس بتاعي اللي هقيس عليه التيومر DNA. وبعدين بالايمج اناليسيس هعرف بقى اوزن الاثنين واقول في الجزء ده من الكروموسوم ده عندي deletion او عندي amplification. Okay, so we come here to the end of our practical session of chromosomal analysis. أخذنا type of chromosomal abnormalities، أخذنا بعدها karyotypes of aneuploidies، أخذنا clinical examples على aneuploidies المختلفة والpolyploidy، وبعدين أخذنا structural chromosomal abnormalities how to describe them، أخذنا karyotype examples على structural abnormalities، وأخيرا أخذنا الميثود other methods of chromosomal study اللي هي اسمها under the type of molecular cytogenetics. Thank you.